Thank you so much for that beautiful introduction, Dr. Chan. My name is Kaelyn P, and I'm a junior at ELNE School taking honors independent research. Today, I am excited to present my research that investigates the relationship between salivary microbiota associated with periodontal disease and Alzheimer's disease. Before I begin my presentation, I would like to introduce you all to Uncle Bob, my grandpa's twin brother who suffered from Alzheimer's disease. Because of him, I gained an appreciation and greater respect for the elderly, spurring my interest in Alzheimer's research and hopefully a future career in geriatrics. Alzheimer's disease is the most common cause of dementia and affects an estimated 5 million Americans. As the disease progresses, individuals experience noticeable degenerative symptoms that impair cognitive and behavioral function. As the most significant factor of Alzheimer's disease and other forms of dementia is age, the elderly population in Hawaii are highly prone to experiencing some form of cognitive decline. The percentage of elderly in Hawaii is one of the highest in the nation, with around 20% of the population being 65 years or older. While the pathogenesis of Alzheimer's has been the subject of extensive research, there is growing evidence that suggests that systemic inflammation plays a pivotal role in neurodegeneration and the development of Alzheimer's. Systemic inflammation can dysregulate the normal mechanism of clearing misfolded or damaged proteins in aging brains, leading to buildup of plaques and disruption of cell function. In addition, neuroinflammation can activate microglia, which are recruited to sites of inflammation and release pro-inflammatory cytokines, further exacerbating neuronal damage. After the gut microbiome, the human oral microbiome harbors the second largest distribution of microorganisms. Current literature demonstrates a link between oral bacteria and a number of systemic diseases, including cardiovascular disease, stroke, preterm birth, diabetes, and pneumonia, which leads to an increased emphasis on evaluating oral health as a source of disease. Persistent low-level bacterial inflammation in the elderly may induce chronic inflammation. In particular, oral bacteria can serve as a source of this inflammation, given the existence of numerous pathways connecting the oral and brain regions, such as through the trigeminal and olfactory nerve complex. The current research includes sampling from intraoral regions such as the teeth, gingival tissue, tongue, cheek, and hard and soft palates. However, sampling from these regions can be uncomfortable with the use of curettes, especially for those with Alzheimer's. Accordingly, saliva covers all surfaces of the oral cavity and collects a wide diversity of microorganisms. Saliva sampling, unlike other intraoral regions, is non-invasive and easily accessible. Notably, saliva also plays a crucial role in maintaining oral health, and age-related changes in salivary function, such as poor hydration and dry mouth, can promote bacterial overgrowth in the oral cavity. Certain bacterial genera are linked to periodontal disease and oral inflammation, such as Perfrominus and Prevotella. This graphic shows the 8 main periodontal pathogens most widely cited in the literature. Due to their high resistance to antibiotics, these gram-negative bacteria may lead to significant public health problems. These oral bacteria are believed to eventually access the brain, leading to neuroinflammation and the development of Alzheimer's. Moreover, studies have found higher abundances of oral bacteria linked to periodontal disease in human brain tissue of Alzheimer's patients compared to healthy individuals, further emphasizing the potential connection between oral microbiota and Alzheimer's. The purpose of the study is to compare the salivary microbiomes of Alzheimer's and non-Alzheimer's individuals in order to establish a more comprehensive understanding of how the salivary microbiome may reflect Alzheimer's status and overall oral health. I recruited a total of 5 participants, comprising 3 from the control group and 2 with Alzheimer's. Among them, only 1 identified as Caucasian, while the others were of Japanese or Chinese descent. Morning saliva samples were collected prior to eating or brushing teeth using the salimetric saliva bio-oral swab. Participants were instructed to place the swab under their tongue for 1 to 2 minutes. DNA extraction was performed using a PowerSoil kit, and the 16S gene was amplified using 27 forward and 1492 reverse primers through PCR. The sequencing was carried out with the Oxford Nanopore MinION, and data interpretation and visualization were conducted through Epitome Viz and Google Sheets. In order to address my purpose, I aim to evaluate differences in the salivary microbiomes between individuals with Alzheimer's and those without. The graph above shows all bacterial genera identified in each of the 5 samples and their relative abundances. The first 2 columns represent the Alzheimer's group, while the latter 3 represent the control or non-Alzheimer's group. The most common bacterial genera identified were Capnocytophaga, Haemophilus, Neisseria, Prevotella, Rothia, Selenomonas, Streptococcus, and Veillonella. I first compared the salivary microbiomes of Alzheimer's and non-Alzheimer's individuals through a principal coordinate analysis or PCOA test, which showed a clear clustering among Alzheimer's and non-Alzheimer's group. This suggests that the overall microbiome differs between both groups. This bar graph represents the relative abundance amongst all 5 samples. Again, the blue represents Alzheimer's and purple represents non-Alzheimer's. Notably, the elevated presence of Prevotella in that Alzheimer's group associated with periodontal disease and oral inflammation could potentially contribute to the observed differences in microbiome composition. Noticeable presence of this bacteria can cause them to invade the brain, which lends credence to the inflammation hypothesis. These results also support past studies that found higher abundances of Prevotella in Alzheimer's individuals. Another interesting observation when analyzing these results was that C3 and the only Caucasian participant had very low abundance of Prevotella. Studies have indicated higher Prevotella abundances in the gut microbiomes of non-Western populations with similar dietary patterns. This dietary correlation may explain the elevated presence of Prevotella in the Asian cohort, providing a link between dietary habits and oral microbiome composition in the studied populations. A low abundance of Peripheromonas was observed in Asian participants in comparison to their Caucasian counterpart. This finding is noteworthy as the bacterial species Peripheromonas gingivalis has been linked to Alzheimer's disease in previous studies. P. gingivalis has been recognized as a significant risk factor for Alzheimer's due to its potential ability to access the brain and activate microglial cells, inducing a pro-inflammatory phenotype. However, well-known periodontal pathogens, including P. gingivalis, were absent in a study on the oral microbiome of Japanese individuals conducted by Takeshita and their colleagues. The absence of P. gingivalis in past Japanese oral microbiome studies may suggest potential ethnic or geographical variations in the oral microbiome, which aligns with the results of this study. In addition, higher abundances of Rothia and Veillonella were associated with the non-Alzheimer's group compared to the Alzheimer's group. Recent studies have established higher abundance of Rothia in non-Alzheimer's group, which supports the results of the study. However, studies have suggested higher abundance of Veillonella to be associated with the Alzheimer's group, which does not support the results of the study. In summary, this study identified an interesting relationship between certain periodontopathic bacteria found in the salivary microbiome and Alzheimer's disease. The PCOA test revealed an overall microbiome difference between both groups, suggesting that the microbiomes of Alzheimer's disease individuals are distinct from healthy individuals. I also identified Prevotella to be higher in the Alzheimer's group compared to the control, as well as higher in the Asian participants compared to their Caucasian counterpart. Additionally, Peripheromonas, which is widely associated with Alzheimer's disease, was found at low abundances except for in the Caucasian participant. Overall, these findings support the claim that the salivary microbiome reflects Alzheimer's status and has implications for ethnicity and diet-focused studies as well. Limitations to the study include a small sample size of only 5 participants, as well as the possibility of sequencing errors and biases. Future research should focus on the role of diet in Alzheimer's development, as well as further investigation into the relationship between ethnicity and the salivary microbiome. Upon reflection of my time in independent research, I acquired numerous remarkable experiences and insights into research that go beyond the typical classroom environment. I developed essential research skills, including literature review, creating research plans, and mastering advanced laboratory techniques. Furthermore, this course steered my future career aspirations, heightening my passion for geriatric medicine and the study of aging. For future research students, choose a topic that you're interested in and don't be afraid to connect with other fellow classmates. As someone who is passionate about Alzheimer's disease, taking this course gave me the opportunity to further my interests in this field of research. Through this course, I also have formed lifelong friendships. The encouragement and assistance from classmates, whether in peer editing research posters or papers, or simply offering moral support, have been immensely gratifying experiences. I also would like to take the time to thank the people who have helped me through this research journey. To Dr. Nishita and Dr. Chalk, thank you for your mentorship and help with recruitment. Thank you to Dr. Chan for guiding me throughout the project development process and providing me with helpful feedback on my poster and paper. Also, thank you to Mrs. Kobayashi and Mr. Hill for helping me with the lab work and analysis. Finally, thank you so much to my classmates and amazing friends Nana, Jolie, Audrey, and Logan for their continuous support. And huge mahalo to you, the audience, for listening to my presentation. Thank you.